Hi guys, so today we're going to be going over the DNA hybridisation process for the OCR A level spec. In this example, we're going to be using the DNA from 2 species, but you could also use it with 3 species. So the first thing we're going to do is we're going to begin by extracting these DNA samples from each of the animals. We're going to cut them into smaller sections. We do this to really just save money, time, and since it's more sustainable, as the larger our strands are, the more energy will be required to break the hydrogen bonds. So the hydrogen bonds we are talking about here are the ones in between our complementary bases in our DNA molecules. We're going to add a radioactive fluorescent marker. It's important that you use those exact words, as the mark scheme specifies that you need this specific wording. But we're going to mark one of the species and leave the other one unmarked so we can identify if our hybrid strands are formed. The hybrid strands will consist of 1 polypeptide from each species. Then we're going to heat our samples. You can either mix them before or you can leave them in their separate test tubes and heat. It doesn't really affect the process. But we're going to heat it up to about 90 degrees so we can break the hydrogen bonds between the complementary base pairs. Now we're going to mix all of our DNA together in our test tube, and we want our hydrogen bonds to reform, so we're going to cool it back down to around room temperature so our bonds can reform between any complementary base pairs. So in this process, when our double helix structures will reform, we can form one of 2. So the first one, we will reform our original DNA strands, and in the second one, we form hybrid strands. We identified this through the radioactive fluorescent marker that we added before to one of the strands. So from here, we can identify which are hybrid strands and which DNA helixes have reformed. But in the test tube, we will have a mixture of both. The main difference between our hybrid strands and our original DNA strands are that not all the hydrogen bonds will form between the DNA bases. This is because as there are differences in DNA, not all of the DNA bases are complementary, so they cannot form hydrogen bonds. So to now test how closely related these species are, we isolate our hybrid strands. Now we're going to heat the hybrid strands in increments. We do this to see how many hydrogen bonds are formed, as if more are formed, we require more energy to break a larger amount of hydrogen bonds. If the species are more closely related, they will form more hydrogen bonds, so higher temperatures will be required to break apart the DNA molecules. And for species that are less closely related, less energy will be required to break the hydrogen bonds as there are less of them. Remember that hydrogen bonds only form between complementary base pairs. So if these base pairs aren't complementary, they won't form hydrogen bonds. So how we needed 90 degrees to break apart the original DNA strands, we won't need that much energy, as not all of the hydrogen bonds will reform. We could have really closely related species, but they won't have the same sequence of DNA, so not all the DNA bases will be complementary. So here we have a diagram of a hybrid strand. So where there are gaps, that is where our hydrogen bonds have not formed. And the ones that join at the centre, that's where our bases have been complementary and they will form hydrogen bonds. In this example, our species are more closely related, because if we compare it to the next molecule that we will see, you will see that there are more gaps between less closely related species. Here we're going to need a higher temperature to break up this hybrid strand. Now this hybrid strand, the species are less closely related. As you can see, there are more gaps in this molecule. Only few of the DNA bases have been complementary. So our non-complementary bases cannot form hydrogen bonds. So here, a lower temperature will be required to break apart our hybrid strand. So I hope you guys learned a lot from this video, and thank you for watching.